Hello, my name is Heather Graves, and I am a program director at the University of Utah Data Coordinating Center. I'm going to talk today about good clinical practice and risk assessment. ICH E6 guidelines for GCP include 8 sections. There was recently some updates in the R2 addendum, specifically changes and updates that came up surrounding the GCP guidelines. No changes to the IRB section, investigator brochure section, and protocol details were made. However, other sections did have some changes. First, let's discuss the investigator responsibilities, namely being able to show investigator oversight in research. It's common to hear no documentation means it never happened. Specifically for audits and on 483s, PI oversight is often a major problem, as well as documentation issues if things aren't organized well. PI supervision is important. Documenting is important. Documenting how other co-Is and research staff are being delegated to is important. We want to ensure that PIs are not asking people to do something they aren't trained to do or that they are not delegated and documented as doing. The other piece is the documentation. Records, electronic or paper, should show what you were doing. Reviewing of adverse events, labs, weekly research team meetings should all be documented through minutes and attendee lists. This shows PI oversight. In summary, have good communication, strong delegation relationships, and organized documentation. Next section to go over is in the sponsor section. A lot of the information in this section pertains to the DCC and lead investigators as we follow these guidelines. They are not only for industry studies, but contain rules and regulations that need to be implemented at the study level. The biggest point in the sponsor section is risk assessment, which is really quality management. Section 5 of the E6 regulations is about quality management systems. The guidelines stipulate that we need to implement a system to manage quality throughout all stages of the trial process. The overall goal of that section is to create a quality management system so that we are identifying risks, mitigating risks, and hopefully reducing major risks that would put the study in jeopardy. We want to think about risk to patient safety, risk to data quality, and risk to study success overall. We want to make sure that we meet study expectations and not fail because we can't get patients enrolled. Overall goal is to ensure human subjects protection and also to ensure reliability of study results by minimizing protocol deviations and increasing overall study quality. The nuts and bolts of this is broken down in 3 parts: identifying the risk, evaluating the risk, and controlling for it if we can't eliminate it. When identifying the risk, implementing a system to manage quality throughout all stages of the study is important. We do this by reviewing the protocol, having conversations in protocol development, and reviewing all components of the workflow to see if there are situations or steps that are going to create risk. We look at what could go wrong, issues that we're worried about, and then have discussions that help us to identify the risks we want to think about. Once we've identified something, we identify how big of a risk it is and focus on study activities that are essential to ensuring human subject protection and trial data integrity. We ask ourselves, is this a critical risk, a heightened risk, or is this a standard risk that we know about and are able to mitigate with our standard processes? These standard processes include using queries, metrics, and reports to help us identify and monitor these risks and creating plans for when we need to take action. This breaks it down even more simply when talking about identifying, evaluating, and controlling the risk, using this rule of 3. What is the risk? Is it a risk to patient safety, data quality, or another type of risk? Is it a standard, heightened, or critical risk? And then we control for the risk. Can we eliminate it altogether? Can we identify these risks in protocol development, something we know isn't going to work at all at the site, and eliminate it altogether from the protocol and replace that process or procedure with something that we know will work and would be less risky? Or is it a risk we can reduce by putting parameters in place to ensure the risk doesn't occur often? Or we accept it and we have a metric or report in place to keep an eye on it. What to expect from the DCC? We are introducing the concept of risk because the regulations say we need to. Talking about risks in grant and protocol development is the first step in identifying those standard and critical risks. Going through the site-specific workflow to evaluate the risks, establish mitigation and monitoring plans, and identifying thresholds is the main part of what the DCC will do. Based on those risks and mitigation plans, reports and metrics, are created and will be reviewed throughout the course of the study. We want to monitor risks throughout the whole study by looking at data and trends. It takes a village to do this, so share information about protocol deviations, risks you've identified, and things that have gone wrong or what you expect will go wrong at your site. What can you do at your site? Look at the processes at your site. Patients are stressed, in pain. Consent could be difficult. Are there processes that are going to be more problematic? 2, barriers to accessing the data. Are you sure you know where all the data elements are in the EMR? Do you see problems accessing the data? Number 3, complicated procedures and developing a plan to address the risk. Are there things you're worried about? Develop a written plan to address it. And number 4, give feedback to the DCC and the lead investigators if there are excessive queries or unclear instructions. This is very helpful feedback for us and helps us as we develop processes and instructions. There are several sources that can help us understand what source documentation is. We're going to pivot a little bit to now look at good clinical practices. The federal regulations have some information that can help guide us, but are fairly vague and complex. We have guidelines which provide more detailed guidance, such as the International Conference of Harmonization and the Good Clinical Practice document. We've also have institutional policies in the form of standard operating procedures that further define what the expectations are. The University of Utah has developed centralized SOPs, and there's one specifically for the collection and documentation of research records titled Study Records Management. This discusses collection and storage of participant research records, regulatory documents, and guidance on providing records for monitoring or auditing. There's also a supplemental document for records that should be included in the participant regulatory binder. The link of where to access these is included in this slide. You can also find additional information or resources by searching FDA regulations and good clinical practice. As part of good clinical practice, good documentation is key. Source documentation should be referenced or placed in the study participant's file to demonstrate protocol compliance and evidence for data entry. If the EMR is not the source document and research staff are recording observations, they must be signed, initialed, and dated by the PI or research team member who's completing the source document. Identifiers should be clear, always use blue or black ink, ensure it's readable, and any changes that are made to the source document should not obscure the initial entry. To ensure good documentation practices, do not use whiteout pencil or scribble. If you have an error, how do you correctly make a change? Correction should be done with a single strike through of the incorrect data. The correct data should be entered with the initials and date of the person correcting, and a brief description of why the error occurred may be helpful. In general, the common practices you should keep in mind are: correction should be made by the original reporter. Correction adjacent to or on a separate form should be made, not on top of the original entry. Never backdate. And never destroy records. Just a reminder that good clinical practice in research is not the same as good clinical practice in caring for patients. Standards for the clinical care of patients is different than standards in academic research and standards in FDA-regulated research. Good clinical practice is a standard for the design, conduct, performance, monitoring, auditing, recording, analyses, and reporting of clinical trials. It's critical that the data are credible, accurate, and that the rights, integrity, and confidentiality of trial subjects are protected. Clinical trials and research should be conducted in accordance with the ethical principles that are consistent with federal regulations and GCP. GCP includes sections that the principal investigator must be responsible for. These include ensuring that the investigators and co-investigators on the study are adequately trained and delegation to others is documented, ensuring that research staff are sufficiently trained on the protocol, and that the medical care of trial subjects is adequately provided and patients' primary doctors are informed. Additionally, the investigator should have communication and approval from the IRB and always be in compliance with the study protocol. If a deviation from the protocol needs to occur to eliminate an immediate hazard to the trial subject, the deviation must be submitted to the DCP. It's also the responsibility of the investigator to maintain adequate and accurate source documents. This means that source data should be attributable, legible, contemporaneous, original, accurate, and complete. The investigator should ensure the accuracy, completeness, legibility, and timeliness of the data reported to the sponsor in the CRS and in the EDC and in all required reports. Any change or correction to a CRF or source document should be dated, initialed, and explained. As was said before, the original entry should not be obscured. As you can see by the examples to the right, this was not done according to GCP guidelines. And lastly, here are some don'ts. Don't erase, whiteout, or obliterate original data entry. Don't accept changes to data without checking the source document. Don't backdate. Don't forget to use IRB-approved consent documents. Don't revise the study protocol. Don't use your staff as subjects. Don't destroy study records. These are all self-explanatory, but vital to integrity of the study and GCP guidelines. Last words, practice GCP and avoid publicity.